Hi, good afternoon. Today we will present about hemophilia. So this is the situation that we have choose. It is about hemophilia that happened to Bob. Haemophilia is an X-linked bleeding disorder caused by deficiency of coagulation. Without these factors, a person cannot stop bleeding when they are injured. Patients with more severe forms of the disease can experience spontaneous bleeding around the joint. Hemophilia divided by two, which is hemophilia A and hemophilia B. In hemophilia A, the missing factor protein is factor 8. And in hemophilia B, the missing factor protein is factor 9. It is caused by different genetic defect. Gene therapy for hemophilia involves using a modified virus to introduce a copy of the gene that encodes for the clotting factor that's missing in Bob. However, gene therapy could offer a one-time treatment. With gene therapy, a function or version of the missing of detecting gene can be delivered into the cells to produce the missing factors. Function of gene need to be delivered into the cells using a vector. This is often derived from viruses, and the vector is entered to only in the liver to therapy the gene. Following treatment with the virus, his should begin producing their own clotting factor normally like in the picture.
I'm Danisha Manikavasan from DBT4B and my metric card number is 23DBT18F2030. The roles given for me are clinical researcher and production specialist. As a clinical researcher, my responsibility is to oversee the clinical trials for medicinal product to ascertain the safety of the product. Clinical trials for gene therapy has no special requirements. The infusion of gene therapy is done through vein in the arm, which is infused during the day, and generally people would be able to discharge on the same day. Once the gene therapy is done, the team will watch this expression of the factor very closely multiple times in a month. Different trials may have subtle differences as to how quickly the factor is being expressed. Monitoring gene therapy clinical trial involve time-consuming for the individual who has received the gene therapy. Depending on the trial, there will be multiple visits to Haemophilia Center that overseeing all the samples collection, which are blood samples, and then also a collection of bodily fluids such as saliva, urine, semen, and stool would be collected. And the important part of the trial is that people need to be aware of. Because unlike other trials, gene therapy trials last forever. Once it's infused, there's no way of neutralizing it or putting it to an end. So the next one is production specialist. As a production specialist, my responsibility is to provide knowledge on strategies of large-scale production of gene therapy for hemophilia. There are few issues on improving gene therapy for treatment of hemophilia, such as variability in responses. Therefore, a strategy to enlarge the scale production of gene therapy for hemophilia is by identifying most variable steps, improve capsid tropism, and so on. Other than that, durability of response. This can be improved by understanding the stability of episomes and degree of integration, transgene toxicity. Finally, high production costs, which requires improvement with scale up. That is all from me. Thank you.